This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 310. Recorded on May 2nd, 2024. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello, and it's a beautiful spring season here with colors popping and grass greening. It's lovely.
Here, it's nice in New York. It's 26°C, which is like low 80s, and sunny. Very nice day outside. Also joining us from Charleston, South Carolina, Michael Schmidt.
Well, hello, everyone. And we have, as they say, Chamber of Commerce weather. It's 82 degrees Fahrenheit. The sun is out and there's no humidity. However, the pollen is out. And all I can say is thank goodness for Big Pharma and their antihistamines.
And from St. Louis, Missouri, Petra Levin.
It's great to be here. We have our humidity is back. Our pollen is back. We have had some rain, though, after a long time, so that's great.
All right. It's time forget the pollen. It's time for some science that distract you from it. And we will start with a snippet from Petra.
So today I'm going to just kind of raise something that actually we haven't really discussed. We talk a lot about the microbiome, and when we say the microbiome, we almost never really define what we're talking about, but usually it's from the colon. And so from the, and the reason most of the microbiome, I mean, there's skin microbiome and others, but mostly we focus on when we talk about the microbiome, we're talking about the one in the colon. And the reason that the colon was sort of the first kind of human microbiome to be examined, as we know that bacteria are very important as our archaea in the colon to kind of help the final step of digestion, but also because to sample the colon, all you really need is poop, which is easy to get. And what's kind of becoming sort of more important now is other microbiomes in the human body. And particularly, I think we're at a sort of new beginning, really starting to look at the small intestinal microbiome. Right, you have your colon, which kind of does a nice U shape, but then you have your small intestine. And when you think about what's inside your gut, you think about your small intestine, and it's, curly and all snaky inside. And it fills quite a bit of space. When they tell you how long your intestines are in elementary school, they're really mostly talking about your small intestine. And the small intestine is the part that goes from the end of your stomach to the beginning of your colon is the small intestine. And it turns out, its own microbiome. And it's been really understudied, in part because it's very difficult to sample, right? You can't easily get to it without some kind of invasive procedure. And people have either had it sampled from the top, if you have some small intestinal problem like an overgrowth or something like that, people will do sampling. But again, that's not something you easily give up for science. And then the other way that has been sampled is for people who've had their colon removed or at least disconnected from their small intestine for chronic bowel disease or for cancer. Reasons are sort of the typical ones. And, sometimes people have their colon completely removed, and then you have this pouch, an ileostomy pouch, or you can actually have one attached at different parts in your colon, essentially to gather the stuff that would normally go through your entire gastrointestinal tract. And so these pouches are connected through a stoma, a little opening, and you can sample from them. And so the questions have been about understanding the small intestine is how do we do it in a way that isn't as invasive and is in healthy people? So once you have one of these pouches put in, you are, in fact, usually healthy. If you've had ulcerative colitis or something, it actually completely cures you because you no longer have a colon for your immune system to attack. So there's been some discussion about it. And so we're two papers that we have posted to the website. One is actually a review entitled Small Intestinal Microbiota from Taxonomic Composition to Metabolism. And then the other paper is entitled, it's actually a paper that came out recently in Cell, Host, and Microbe, and it's a research paper or resource. And it talks about the plasticity of the adult human small intestinal stoma microbiota. And really, it's a resource, meaning it should be of use. It sort of presents things that are useful for investigators going forward, how to study the small intestine, because it does some nice diversity studies, sort of what's the taxonomy in the small intestine, what's the diversity in the small intestine in people who are having these ileostomy pouches and pouches at different points in the colon put in the surgery because they sample before and after surgery, and then sort of longitudinally as long as from these pouches that are on the outside of the body. And so I thought it was just worth discussing, not too long, but just briefly, kind of the small intestinal microbiota, because actually most of your food absorption happens in your small intestine. And I actually learned a lot reading these because I hadn't thought that much about it, because the review really talks about how the small intestine has been difficult to study, which I mentioned, but also it's a very specialized place because it has a huge range. For example, the pH at the top is very low because your stomach pH is very low. And then at the bottom, it basically comes closer to neutral or maybe even slightly basic as it gets towards the colon. So you have a big pH range within the small intestine. You also have, you go from a primarily aerobic environment to an anaerobic environment, or a very little oxygen as you get near the colon. So you've got this big diversity. You also have all these secretions, your body's secreting all these pancreatic enzymes to help you digest things. You absorb sugar and amino acids, among other things, in the small intestine. And so you have, the bacteria there are really important because they're helping digest your food a little more, break it down. And so I thought that was really interesting. They talk about the different methods of sampling in detail. I forgot, actually, there's this new method, and we've talked a little bit about this. I always think of Inner Space with Dennis Quaid, these movies, but they have these capsules which people can swallow, and they sample. They are open in your small intestine and then somehow close to sample. Obviously, then you have to retrieve them from poop, but they're not working perfectly, but that's another way to get samples. So I think this review by Simon Yersin and Pascal Bonesh is really great, not only because it describes sort of the methodology that people use, but it has these really wonderful figures. And figure 1 is excellent. It talks about the core microbiota and in the small intestine, and there are really 4 groups in there. There are sort of gram-positive bacteria, actinomycetes, some gram-negative bacteria as well. So they say the core microbes are Streptococcus, Veillonella, Fusobacterium, Prevotella, and Haemophilus. But they really sort of are striated throughout different regions of the small intestine. So they're almost like in a continuum, but sort of like different segments have different bacteria. And this makes sense because, first of all, the pH is changing, the oxygen amounts are changing as you go through the small intestine. And they also show kind of what is in the small intestine that comes from both the host side, so like lipases and peptidases and pancreatic enzymes, as well as kind of the ions that are in there and vitamins, which also influence what the community is in there. So again, I think this review is really useful. So again, I think it discusses all these different ways to get at the small intestine. But this review is a little older, and it says that they're not sure that these ostomy bags are really, reflective of what's normally in there. And so that was kind of a big thing hanging out there. Like, what are we getting an accurate view? Are these 4 phyla, the Bacilliota, which used to be the Firmicutes, I'm only now learning that they've been renamed, the Bacteroidota, which are formerly the Bacteroidetes, the Pseudomonads, and some of the fusobacteria, and the actinos, are these guys really what's in there? And that's why this paper by Ilmaz, Führer, Morgenthaler, Krupka, Wang, Sparry, Candinas, Misselwitz, Belde, Sauer, and McPherson, I think is really great, this paper from Cell Helps Micro, because the first thing that they do is this controlled experiment to show that what's in these pouches is actually the same as what was in the intestine beforehand. So they have in their first figure how they do this experiment, sort of where these pouches are, sort of the stoma, the sort of openings are into the intestine, and how they do the experiments. And so that's excellent. But then in figure 2, they really show that the composition in the ileum before surgery is not very different from what's actually in the pouches after surgery, even many, many days out. And so it sets up that you can use this less invasive method to study what's in the small intestine. And again, this is critical for understanding what's there.
And could I just say, I just think it can't be understated what a bold plan this was to take advantage of this patient population, to collaborate with the clinical side and do really extensive analysis over time before and after eating, et cetera. It's an impressive data set that they generate.
It is really incredible. And if people are interested in studying, first of all, this resource and their methodology, the methodology alone is super useful, but as a resource of what's there and not there is also really interesting, because they show there's very little in the way of monosaccharides and disaccharides by the time your food moves through your small intestine, which says that it's all absorbed. And they show that the microbiota, the sort of big picture composition at sort of the group level, is pretty stable, but some of the subspecies fluctuate when you eat, which makes perfect sense. You might have a bloom when you eat because now there's more for them to eat. And I thought that was just really interesting and exciting.
And it'll be a theme that we carry on in the next paper in the desert.
Oh, yes.
Exactly. I mean, this is like
food,
when you
the bacteria are like snap to attention.
Exactly. They snap to it. And things move very quickly through your small intestine. So if you haven't eaten for 8 hours, it's pretty much clear.
Or it's like cookies in the coffee room or blueberry muffins in the coffee room. All the graduate students flock.
And some of the faculty.
That too.
But again, this is just fascinating because we haven't really thought about it. We don't talk about it much. And so between this review and the paper, I thought it was really interesting. One of the kind of maybe not surprising things that I learned, but for some reason feels impactful, is that people sometimes have bacterial overgrowth in their small intestines. It's very difficult to get rid of, but it comes from 2 places, which again is either your oral cavity, your mouth and your pharynx, and in which case it's mostly streptococci, or it comes from your colon, in which case it's mostly enterobacteria. And so it makes perfect sense that that's where the overgrowth is coming from, because they're not supposed to be there, as many of them. And that seems to be your 2 choices, but to actually know that's what happens is very satisfying. So
and if you look at what the small intestine is processing, it's processing primary food that we consume. And so it's liberating and then making waste products. The large intestine effectively takes the waste products that are produced by the microbes in the small intestine and then uses it to make more biomass in the large intestine to effectively dewater and grab any remaining nutrients out of the food that wasn't grabbed by the small intestine microbiota. And the consequence is we're very efficient, which is principally why we're all getting so fat.
But also, your food doesn't spend very much time in your small intestine, so it has to go very quickly. And your large intestine is fermentative; it takes much longer, 8 to 10 hours. So that was also really important. And the other kind of cool thing that I learned is that, remember, we eat our amino acids. Getting complete protein is a really big deal, but the bacteria that are in our small intestine, right, some of them are excreting amino acids and other things. So we can absorb from them some of these amino acids. And some fibers or some like more soluble fiber and things are actually broken down by the bacteria in your small intestine. And then the other stuff, has to go through the more sort of complicated fermentation process in your large intestine. Anyway, I thought the review was really interesting. And I thought this paper was just great. And it's just like a whole new world of microbiology, which is, of course, awesome.
The other impressive thing about the paper is they could say that in general, the community in the small intestine was stable, like who is there was fairly stable, even though after fasting, there weren't many microbes there, and then they bloomed. But then they looked in more detail, and they found that within a particular strain, there was actually quite a lot of genetic variation as judged by sequence variants. And so some would bloom, others would be lost. Overall, that strain would be stable, but there was constant selection probably, and for particular ones. So just amazing evolution happening on small scales.
Right. It's almost like a chemostat. It moves so fast.
Yeah.
So it's constant culture. It's basically constant culture, except for in a chemostat, you want to keep your nutrients. So a chemostat is how you keep bacteria. You sort of add nutrients and dilute it. So the bacteria always stay at the same concentration. So when you're eating, though, obviously, it's more like you're giving your bacteria in your small intestine a ton of nutrients, and then you starve them, and then you give them a ton of nutrients, and then you starve them. So it's a very different feast-famine situation. And again, in your large intestine, it's much more stable. So
they also, Vincent, really had some evidence from sequencing that there are bacteriophages in there that were also contributing to who blooms and who doesn't. And so it might look in some of the graphs as though things are fairly static, but there is a lot going on at the substrain level and microbe to microbe.
Yeah, I also found it comforting.
I was waiting for Vincent to comment about where are the viruses?
There you go. There they are.
Yep.
Because I think, we're only beginning to understand the microbes in the small intestine to a first approximation. And I think we really need to think about the whole lysogeny lytic phase of phage that are going to control the population dynamic that we're seeing. And similarly, there are phage in the large intestine as well that are contributory. And I don't think anyone has actually looked at the contribution of the virome in the context of the microbiome. Mm. And when people say microbiome, they don't often consider the virome, which is completely distinct in most people's minds from the microbiome, which we only consider to be bacteria. And occasionally a few people will throw in the fungi, namely the yeast that can live in our guts.
Here they just looked at DNA viruses because of the extraction method.
Right.
But,
yes. Right.
But they found that for those, it didn't change much with eating. So it's a pretty stable viral population.
I think it should also be comforting for people who have these pouches that essentially they're not changing your microbiome very much. Mm. And, my understanding about people who've had these surgeries is, especially if you've had ulcerative colitis or IBS, something very painful, that essentially you're cured by this. And so it's good. You can be very healthy and your microbiome can stay healthy.
A couple of the resources that make this kind of culture-independent research possible, they use the Human Metabolome Database, a public resource, and also the Metabolomics Innovation Center has a different one called Food Database, FOO. It's kind of a play on words, FOODB. So the whole community can now go into these data sets and begin to deduce what is happening, who's there, what's happening, based on their DNA analysis, culture independent.
All right. Thank you, Petra. All right. Now it's Michael's turn.
Yeah. So we're going to go to a different part of the planet, so to speak. We're actually going to go into the desert. And the title of today's paper is Survival and Rapid Resuscitation Permit Limited Productivity in Desert Microbial Communities. And it's a paper by Imminger, Meyer, Schintelmeiser, Levin, Schnecker, Richter, Gilor, Eichenhorst, Wolbakken, and it's an open access paper that appeared in Nature Communications in mid-April. And these folks are at the University of Vienna in Vienna, Austria, and they're in various departments throughout that university. They're also affiliated with the Ben Gurion University in Israel as well. So microbial activity in drylands tends to be confined to rare and short periods of rain. The microbes in this niche live in the top of what is termed the biocrust, the top millimeters, and that's the biocrust, where they tend to enrich the soil with carbon and nitrogen, while at the same time they facilitate the prevention of soil erosion and the retention of what little water the crust, or if you will, soil in the drylands can possess. Unfortunately for our ever-growing earth-based population of humans, drylands now constitute 46% of our terrestrial surface, and they play an extremely important role in our global carbon cycle. So it's not surprising that drylands are currently expanding due to global climatic changes. Now, the operational definition of what a dryland is what you might expect, that they are fields subject to long periods of drought with rare and short periods of rain that may only last a few days in the whole year. So you consider places like the southwestern United States as an example, or as in the case here, the Negev Desert, where the word Negev is derived from a Hebrew root denoting dry. And so the Negev Desert, I'll put a map into the show notes, is actually in the nation of Israel. And one thing that I had not considered when I first read this paper was the activity of desert microbes is largely confined to the short windows of unpredictable episodes of rain, and thus so the main microbial mediated ecosystem processes associated with drylands or desert soils is subject to this stochastic process of when is it going to rain?
And a lot of patience.
And as the authors offer in their introduction, it remains unclear how microbial functions are executed if a considerable proportion of desert soil microbes remain dormant after rehydration, or whether they all reanimate, or if they exhibit considerable delay in their response to the rehydration, as was often reported for other semi-arid soils. Or if you will, the outcomes that one would measure, namely the fraction and identity of the responding desert soil microbes, their census, that is, who's there and how many and what they're doing, and the associated resuscitation speed of the population. Now, we cannot forget that growth is a population thing. You're measuring the change in population, not in the ability of the individual to divide. We're looking at the population's ability to divide. So
classically, yes, that's what we're looking at.
Yeah, classically.
But this is very modern.
So in other words, we should consider that rehydration offers us an opportunity for rapid growth, which in turn should be the key to the maintenance of ecosystem processes in such a narrow and unpredictable activity window, which is then only further complicated by the consequences of desiccation and then the effect size on the community and or individual that rehydration has. Simply put, what does osmotic stress do to the biocrust population? For it has been suggested that dry down and or rapid rehydration of soils via rain can cause cell death. And anyone who has ever lyophilized a bacterial culture know the great lengths we go to preserving the dehydration of microbes when we lyophilize them. So, but that's a story for another day. Well, they certainly have teed up many issues for us to think about in their introduction, especially since we're seeing such a growth in the area of drylands across our planet. So how did they approach the questions? Well, here they used simulated rain, but they didn't use any rain. They used deuterated heavy water. So they could employ a new technique that I don't believe we've ever covered on TWiM. And this technique is termed single cell, nanoscale, secondary ion mass spectroscopy, or NanoSIMS, which is nothing more than a special mass spectrometer with special optics that creates incredible spatial resolution, where it enables the observer to witness cellular processes occurring within a 50 nanometer window, so they could study the structures in the processes carried out by a single cell and literally watch an individual work. And that's because of the deuterated water. They're looking for where the hydrogen is going in the materials that the cell is making. But that's not all, folks.
And
Go ahead, Michele.
Could I interrupt? This
Yes,
NanoSIMS, as you point out, was a really cutting-edge technology. And I just want to the author's credit, this was not just sitting on the shelf. They didn't just pull it down and hit the on button. They went through many iterations looking at the literature. What methods can we apply? They tried trial and error, trying to look for something that would be robust and would be applicable for this population. And so the two authors went through very frustrating periods of optimization and testing. And Dimitri, one of the first authors, called it an odyssey to measure this single cell biomass generation. But in the end, because they tried Raman spectroscopy and another method, but they just wouldn't work in the soil crust system. So they finally landed on NanoSIMS, and you're going to show us what beautiful results they got.
I mean, just jump to, this is open access, folks, so you can actually go and see all their beautiful pictures. This is what's in figure 2. If what Michele just tumbled to is figure 2. But that's not all that they used. In addition to using the NanoSIMS technique, they also employed metatranscriptomic analysis, where they were able to show that the biocrust microbial communities from the Negev Desert were found to have limited productivity. That is to say, they made only certain things with median replication times. And now here we're measuring the population effect of between 6 and 19 days, with a restricted number of days that allowed for growth. And as another one of the first authors commented in one of the press releases, and this was Stephanie Imminger, offered that they found that almost all the biocrust cells reactivate. And this is what really shocked me, that they reactivate. But with the limited rainfall, only a small proportion of the cells would be able to double. So they didn't put their energy into dividing. They put their energy into effectively saving up for the next drought.
Repairing any damage, DNA damage,
repairing any DNA damage. And the way they figured this out is they used genome-resolved metatranscriptomics. And they had already previously characterized this desert population. They did the metagenomics, so they knew who was there. And then they threw the transcripts against this data set to effectively allow them who was making what, when, and how much. This allowed them to demonstrate that nearly all the microbial population was able to resuscitate themselves within minutes after seeing this deuterated water using, and it was found that this was, and this is what blew my mind, was independent of the taxonomy or the microbes present in this desert community. And as you can well imagine, there were a lot of cyanobacteria, which are blue-green algae, in this desert population, taking advantage of the feast and famine of the water, and they're able to photosynthesize. And that's a bit of the story of how they can survive the hard times, because they can steal light energy to effectively make ATP. But what they're up to in this paper is they're investing. They're learning how the microbes are investing this precious water into, as Michele already said, the repair processes, making certain that their genetic material was up to snuff, and energy generation. They're effectively making storage compounds that will get them through the cold, dark winter. If you will, they plant a garden, and then they effectively put up what they've grown, and they put it on their pantry, so that in the cold, dark winter, they can pull from the pantry and survive until the next rainfall.
Or the hot, dry winter, as the case may be.
The hot, dry winter. So, if you will, offering that these biocrust microbes make optimal use of these limited windows of opportunity available to preserve the individual rather than the population until literally that rainy day. Now, the data that they offer us.
Michael, I had a question, which is, I mean, on the one hand, yes, I mean, it makes sense to me that it's an individual, because they would have to have some way of communicating if it was some population level thing. But then also, what is the density of these guys in the soil, in the crust?
They're not very dense.
So, I mean, they can't really quorum sense or anything.
No,
this has to be,
There's no conductivity. There's no conductivity. And in fact, many of them are cyanobacteria. And as you know, cyanobacteria effectively form long filamentous chains. The other major players in this population of microbes are actinomycetes or actinomycetes. And so what they're focusing on is, I think they're beginning to try to let us understand what's going on in these very dynamic and stochastic processes. And they develop data that are along four major themes. First, the majority of biocrust cells can resuscitate in a simulated rain event, albeit with slow growth rates. And I'll get to the data that support that. The second thing that they focused on, and again, this is what Michele tumbled to, is the microbial populations in this feast and famine situation from available water generate energy and repair DNA at the onset of resuscitation. And that was what was found to be so critical, because in these long extended periods of drought, they're generating free radicals from the normal consumption of the stores of energy that they may have maintained to maintain their minimum energy charge. The third area is that, and this is really pretty cool, is microbial populations acquire carbon and energy during the main hydration phase, not the initial phase when they effectively are reanimating. If you will consider the rain being the delivery of the monthly check. And so what the microbe does before it goes out to the bank to cash the check, and microbes are all cash society, there's no credit. They first need to cash the check. And before they do that, they have to burn whatever energy stores they have on the shelf. So they are showing us through the transcriptomics what the microbes are consuming in that initial rehydration phase. And then they go shopping to restock the pantry and, if you will, fix the car, namely the DNA repair. And finally, the last bit, and this is perhaps, I think, the most important bit that I found to be most intriguing, is the population was protected by the accumulated antioxidants, and that they acquire energy from inorganic sources, namely because a lot of these are photosynthetic microbes. So they're able to effectively get their energy or consume energy in the dry phase. And it was all done with a combination of this nanosims process and the transcriptomics that they did. And for this last point, before I go back to the beginning, the transcriptomics found with the reactive oxygen species in energy generation from inorganic sources showed distinct patterns, and they were particularly abundant in the dry phase, which was at 0, 39, and 55 hours, and were found with higher proportions of manganese-based catalase transcripts in the dry phase. And manganese is, of course, the transition metal that's involved in catalase, which can effectively defuse the molecular oxygen grenade that goes off just from normal interaction with oxygen radicals in the environment. And this wasn't in just one microbe, but it was in 37 metagenomes that they evaluated. So it was pretty cool. So now, looping back to the first data bullet, that the majority of the biocrust cells resuscitate in this simulated rain event. And this is where they're using the nanosims, using the deuterated water. Here they simply ask the question, where does deuterium go? Learning that the cellular isotope enrichment experiments, and this is not for the faint of heart, these are challenging, revealed that in almost all the biocrust cells that were retrieved with the applied cell separation and concentration approach, the anabolic pathways were reactivated by the rain to a level sufficient for biomass production, which is shown in their first figure where the Negev desert biocrusts were exposed to the simulated rain events. And it looks like a reverse growth curve. They get rehydrated, and then it slowly asymptotes down to where there's effectively no metabolism. But when reactivated with the heavy water, the microbes can covalently bind the deuterium, mainly in the carbon-hydrogen bonds during, wait for it, de novo lipid synthesis via NADPH. And thereby they incorporate the isotope tracer. And the hydrogen isotope concentrations, they have beautiful figure too. It just goes on, show the compositions of the biocrust filaments of the cyanobacteria and the non-cyanobacterial single cells and the resulting deuterium compound. They give us a breakdown within the first 3 hours of hydration. 68.4% of the single cells were significantly enriched with deuterium and thus considered active, reaching 91% reactivation after 12 hours.
It's amazing.
If we dehydrate for less than 8 hours, we're in trouble, folks.
9 to 10 are dead, not up and
we're
kicking.
falling all over ourselves.
Spend some time on figure 2. For the sake of time, I'm not going to go into all their beautiful data. And it really is so beautiful. The take home is you need water to divide. And I think even a first year undergraduate would say, well, that makes sense. But their next experiments support the claim that rapid reactivation of diverse microbial populations occurs almost universally upon rehydration. And this, again, is from the beautiful metatranscriptomics data from their rehydration experiments in which the biocrusts were exposed to the simulated rain event that hydrated the samples for 24 hours, followed by a desiccation phase. And looking at the transcripts. So now they look at transcripts from 15, 30 minutes, and then 3, 6, and 12-hour hydration time points, and at time points 39 and 55 hours. So you get a sense of what's going on in this desert. And what they learned is that 2 phyla recruited the majority of the transcript or produced most of the transcripts. And those were the cyanobacteria and the actinobacterioida. And among the analyzed hydration time points, there was no systematic or taxonomic shifts. I mean, everybody was rehydrated. They were all in the pool together, and they all came up. And
you snooze, you lose.
Yeah, I mean, everyone plays this game, folks. It's not, I'm going to wait for Norman to take over. I mean, they did everything. And then they did something really clever in that they normalized the number of transcripts per metagenomic assembled genomes, which then enabled them to investigate differential expression of genes between the different time points and phases of the experiment with individual microbial populations.
So this is a lot of computational biology.
This is not for the faint of heart.
The lights are dimming as the computers are on.
I mean, the most significant changes in transcription occurred, as you might guess, between 0 and 15 minutes, 30 minutes and 3 hours, and between 12 and 39 hours. As if you think about the growth curve, it's effectively going through lag, it's then going through log, and then it's going into stationary phase. It's really pretty cool. And they go into summing up this data bullet through their use of this combination of genome-resolved metatranscriptomics and single-cell activity analysis, which is presented beautifully in Figure 2.5. You can be a firsthand witness to the wonder that is microbial resuscitation dynamics, and then begin to appreciate the underlying molecular mechanisms following simulated rain events in a diverse, arid biocrust microbial community.
And again, this is all culture independent, right?
Yes.
They went out, they got the samples, timed it just right, sampled, the same depth. They had to get very uniform depth and size of the samples for.
There are a lot of controls we don't have time to go into, but it's a beautifully illustrated and documented paper. The next aspect of their study, the one that I metaphorically referred to as spending your paycheck as soon as it is cashed during the early days of the month, they provide us data that supports that metaphor, that the microbial populations generate energy and repair DNA at the onset of resuscitation. So, if you will, they they take the stock out of the pantry and fix the car. And here they showed during the early hydration event, in that 15 to 30 minutes after the addition of the deuterated rain, the transcripts for DNA repair and energy generation were significantly increased. The transcripts involved the repair of double-strand DNA breaks, which is common to dehydration during desiccation. And they were more abundant across numerous taxonomic groups. And they give us they give us Rubobacter, Chloroflexuri, Cyanobacter, the Bacteroida. They go through all the lists. One question I, though, had is, where does the energy for DNA repair come? Well, they offered that the energy for DNA comes from the pantry. Namely from the degradation of storage compounds like polyhydroxyalkanates, which, if you remember your secondary metabolism, that's where those suckers are made before bacterial populations go into stationary phase. Well, the microbes make that, and then they put it on the shelf, and then when they come out of starvation, they have to eat something, and they're effectively eating that can of corned beef hash that you bought and you never knew why you bought it. But it's really quite beautiful how they show that this PHA is gone. And the way they did that is they looked at glycogen storage. Now, glycogen is much easier to eat. It's sort of like the bag of candy on the pantry. That gets gone first. And the glycogen gets consumed, but it's not made into the middle of the hydration phase. So there's no sense in making a transcript that can degrade glycogen until you have some glycogen. And so it's really quite an impressive study. And then the transcriptomics then also shows us that organic compound transporters revealed that their highest transcript abundance was in the main hydration phase, where they're bringing in the building blocks, suggesting that organic energy sources simply play no role in that early resuscitation mechanism. And they speculate that it's because the majority of these creatures are photosynthetic. They're making their energy gradients or proton pumps via using light. And the third bullet, that the microbial populations acquire carbon energy during the main hydration phase, gets us to the concept of growth or population. And here, this is reflected in the transcripts produced are mainly for energy generation and carbon acquisition. And, when you get to all of the things that they did, you really find they did a remarkable job. But the one thing that I found to be most inspirational is the microbes know that dehydration is coming. And that, of course, was designed by natural selection. They don't know anything. It's just the selection.
Experience has
yes,
taught the
experience.
lineage.
You adapt or you die. And that's the fourth data bullet, is the microbial populations are protected from accumulated antioxidants and acquire energy from inorganic sources during this dry phase. And again, this was based on the transcriptomics analysis. And they put all of this together for us in a summary figure. And walking, and that's figure 7, and walking across their figure from left to right, we start in the drought, or the dormant phase, as they call it. Their reactive oxygen scavenging. They describe that the osmoprotectants are present, and they're effectively oxidizing hydrogen. Hydrogen is a great energy source, and they're effectively able to oxidize hydrogen. Then we get to the early hydration phase, or as I refer to it, eating the pantry staples through the consumption of the PHA. Also, the light-driven ATP production is here, and they go into talking about bacteria rhodopsin transcripts, which is involved in the halophilic archaeobacteria. And of course, the halophiles are a hallmark microbe in the Negev desert, and I think that's where they were first isolated. And again, bacteria rhodopsin is probably one of the simplest proton pumps known, and it just it effectively sees the light, it undergoes a conformational change, and that effectively pumps the proton. And then finally, during the main hydration phase, they observed the glycogen degradation and carbon acquisition. Hydrogen oxidation continues. You get chlorophyll synthesis so that you can continue to make some energy from the light. You may not have a lot of water, but at least you have it, and then they make the osmoprotectants. Putting this all into perspective, there's hope for a drought-filled earth, I guess, as long as you're microbial. Not much hope for us, but the microbes have learned how to adapt to these stressors, and they experience it in deserts, namely high temperature and radiation, and they have these osmoprotectants built into their genetic code, so that they're able to survive these extended droughts and infrequent and stochastic sudden rain events, which was all supported, again, beautifully by their nanosims data and their metatranscriptomics data, which in essence offers that the microbes in these dry lands have adopted and adapted a strategy where they are relying on their ancestor or their previous generation to have protected them from the oxidative damage, and through the accumulation of all those important trace metal manganese compounds coordinating the catalase. So, they're ready for desiccation. So, Michele, I know you've talked to the
I
author.
have, but actually I want to just contrast the strategy of these desert microbes in the crust to what we learned classically in microbiology, that the hardy microbes that can live in the soil for a long time were like, spore formation. That's the strategy many microbes use. Well, these microbes do not have the luxury of undergoing that cellular differentiation. So instead, they've got a much more streamlined strategy of like osmoprotection, turning on, turning off the cell cycle.
No, I found this super interesting because we talk about osmoprotection, but maybe actually it's really a drought rehydration retraction system, because for bacteria, right? I mean, E. coli goes through lots of changes in osmolarity, but for a lot of bacteria, the osmolarity might be relatively, but they're going to dry out. Even bacteria in hospitals, right? You have bacteria that survive on surfaces completely dry, and that's a big contamination problem. So, I was thinking like maybe we've defined osmolarity wrong.
I think this study really gives us a lot to think about.
Yeah,
And
absolutely.
I also makes me think about what we might find as we explore deeper regions of space. Because if these microbes can persist so efficiently that 96% are reactivating right when there's a little bit of moisture, surely we're going to find something interesting on Mars or the moons of Jupiter. Yeah?
Just give them a little water.
At the right temperature, that's the problem.
Yeah.
It's so cold.
What's interesting here is that they're, as you say, they're well adapted, but dry lands are expanding, as they point out, and the microbes there are not going to be able to adapt so quickly, and that could be a problem.
It will be a problem, especially from the agroeconomic perspective of being able to grow food.
So there are a pair of first authors. Stephanie Imager is one of them. She's currently a PhD student in the Vupkin lab, and she is an avid listener of TWiM and was very honored that we are following her podcast, and says she enjoys listening to our discussions and always learns a lot outside of her field. So she became fascinated in environmental microbiology in her early years at the University of Constance in Germany. Her lecturer, Bernard Schenk, really got her fascinated, and especially she got interested in the question, how do microorganisms survive harsh environmental conditions? So she did a master's at the Swiss Federal Institute of Aquatic Science and Technology in Zurich, and was actually studying microbial resistance to dissolved copper ions.
Cool.
She then stayed on and worked as a research assistant looking at transmission of antibiotic resistance genes in waste and drinking water. And during that time, she went to a conference it was the 4th annual How Dead is Dead conference.
Cool.
And that's where she met Dagmar Vupkin.
I had no idea. That is an awesome conference.
Isn't it great? So that's where she met the senior author of this paper, Dagmar Vupkin, who is now her thesis advisor at the University of Vienna. So she did say that the sampling in the Negev desert was exciting, but it was incredibly difficult. They had to do a lot of trial and error and troubleshooting before they got to this step of all this beautiful data that they eventually generated. She grew up in the Alpine region in southern Germany and enjoys hiking and mountain biking or just relaxing at the lake in her free time. So she partnered with Dmitry Meyer, who's now a senior scientist at the Department of Ecological Microbiology at the University of Bayreuth in Germany. And he got his start first studying classical micromolecular biology, but then realized that environmental microbiology is like a whole nother unknown and wanted to really focus on that. And as a master's student, he had the opportunity to go on a research cruise. And from that moment, he was hooked. He then went to the MPI, where he studied marine microbiology in Bremen. And there he studied deep sea hydrothermal vents, which was exciting, but it's not often a student gets to go on one of those deep diving robot trips to collect samples and do all that. So he realized he wanted to find an experimental system of environmental stress where he could have a more hands-on approach. So the field trips to Israel, he said, were a lot of fun, but hard work collecting the soil and crusts. And again, none of them had experience doing metatranscriptomics in soils, and he was skeptical, frankly, that they were going to be able to figure this out because the nucleic acids are hard to extract from soil. There's a huge diversity of organisms in the soil, so you're going to have very complex data sets. And also the soils themselves are heterogeneous, so you've got to be really careful about where you collect your samples in a time series. So they were thrilled when actually they were able to extract deduce very clear patterns of the life cycle of these microbes. He also said that they generated so much data, they actually published a companion paper that prelude to this in 2021 in M Systems. So if you want to look at more data on this, you can find it there. And they've generated, of course, a multitude of new hypotheses from this data, and he's looking forward to now following up on that work in the years ahead. So he says in every new environmental experiment, you've got to optimize the methods. You can't just jump right in, pull something off the shelf. Likewise, publishing the paper was a challenge. They were striving to publish it at a place that will get a wide readership, which means there was some back and forth with the journal and et cetera. But once they finally did get reviews, he said they were really experts in the topic. They really understood the study, made some great suggestions, and now that the paper's published, they're really proud and delighted that they're getting such a great response. Dmitry also points out that he was working on another project on hypersaline microbial communities, again, a water-limited environment on the Arabian Peninsula. And one of his colleagues was a Palestinian. So traveling and going across the border was interesting. He frequently got questioned, who do you know? Who are you going to work with? So he'd constantly say, I'm a microbiologist. I'm visiting other scientists. We're studying nature. And Dmitry hopes that one day we will live in a world where this won't raise any suspicions. He really enjoyed getting to know both Middle Eastern cultures and met very smart and kind people in both places. And it really hurts him to watch the current conflict that we're all aware of. Dmitry's partner is also a scientist, so they have a family. They're trying to do the dual career thing without having to, live in separate towns. They're optimistic that they've landed in a place that's going to let that happen. He also really enjoys field work, as you can see, and as a hobby, really relies on music to keep him going through all the ups and downs. Likes to go to live shows, playing music at home and in bands, and just listening whenever possible.
Well, Vienna's a great place for music.
Yeah, depends what kind of music you like.
Well, if you like Mozart, it's fantastic.
Yeah.
If you don't like Mozart, I don't know. Maybe the Grateful Dead.
There's probably a good scene.
Yeah.
They have excellent
he also points out
too.
that through his career and moving, with his family from among different institutions in different countries, that he now has friends around the world, and that's something he really treasures about being a scientist.
What were we going to say, Petra? You got
I was just saying Vienna has excellent pastry.
They do.
Oh, yes.
They do.
Coffee and pastry, no better place.
Yeah, Dmitry pointed out that he and Stephanie first met at a delicious bakery over pastries, kind of started to brainstorm this project. So that the Vienna pastries contributed to the work.
They are inspirational.
Thank you, Michele.
Yep.
And Michael, thank you. And Petra, thank you. That's TWiM 310. Show notes at microbe.tv/TWiM. If you enjoyed this kind of discussion of science, please consider supporting us financially. You can go to microbe.tv/contribute. We are a nonprofit entity, Microbe TV, that is, 501(c)(3), so your deductions are tax deductible from your federal U.S. taxes. And if you have any questions or comments, TWiM at microbe.tv. Michele Swanson's at the University of Michigan. Thank you, Michele.
Thank you. My pleasure.
Petra Levin's at Washington University in St. Louis. Thank you, Petra.
Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.